we've been working on kind of rebooting the BEL platform, and the name that we decided upon was BEL.bio because we found out that the domain name was available and it was a really, really easy one to remember. It's just essentially become the name of the platform. So whenever I talk about the BEL.bio API, I'm talking about this particular reboot. And one of the things I just wanted to throw in front of you, and this is part of the advertising and marketing, is how do we communicate to people what BEL is very quickly? I'm still looking for refining our elevator speech of, to me, BEL is for biologists what the chemists have had in the chemical reaction language. And there's actually no name, as far as I can tell, for the set of things that chemists use for understanding chemistry. So I just call it the chemical reaction language, and that seems to resonate. But if you have any ideas, and hopefully we can talk about this over lunch or continue to have conversations, what is the best way to get across to biologists who think in networks, and everything in biology is about the network, and most of their communication, most of the real communication really is sketches of network diagrams or signaling pathways. How do we get them to understand that BEL is their language? So that was just kind of a put the thought in your head so we can start thinking about it. For BEL.bio, after losing Nick Bargnesi and Tony Bargnesi over in the corner, and having more of a team to support a more enterprise-focused OpenBEL API, I kind of had to go back and think about what was maintainable in a smaller setting as well as more community-friendly. Because the platform we had, I had thought that Ruby with its metaprogramming would kind of make a lot of sense for a DSL domain-specific language like BEL, but very few people want to learn Ruby in order to work with BEL. So we decided to revamp it and essentially recreate it using Python. Docker had matured greatly at this point, and we rebased on top of Python and Docker. Some things that we really wanted out of this, and this is the third time that we've essentially built a parser validator. The first time the BEL completion service was semi-functional. It kind of helps you figure out what functions or namespaces you might want to use, but it was kind of disconnected. The second one that Tony built was much better, much more usable, but there were still some mismatches in what we wanted to do and what we were able to do in that platform. So this time around we were focused on providing as much support for the BEL statement or BEL assertion creator as possible, and make it as easy, because BEL isn't super hard, but there's still an activation energy to get people to use it. We wanted to make it as easy for people to use as we could, and we want to continue to make it easier and easier as we go along. We also wanted to provide kind of basic platform API services, like we started with the OpenBEL API, but really make it both library as well as API services as well as command line interface, that kind of no matter what you wanted to do, we could provide support. And now, in the progression of talking about BEL, we've had a lot of different terms that have shown up in our lexicon. We've talked about CAMs, knowledge assembly models, and BEL statements. We actually went from annotations, Natalie help me here, to experimental context with the OpenBEL manager, and now we're back to annotations. We've talked about BEL statements, we've had BEL evidences, which was what we're now calling nanoposts. So there's a lot of different terms in the lexicon, so I just wanted to start off and say we have, with as much community involvement as possible, discussed it And BEL assertion seems to be the best way of talking about BEL statements, because they are assertions, they're not fact. A statement has a little bit more context of being a fact, and as Jamie at Patients Like Me said, you know, these are assertions. These aren't necessarily real until they've been further validated. We also talk about the BEL assertions as either a single string, which is often how you see it, or as an SRO, subject, relation, object. The BEL nanopub comprises everything about a or several BEL assertions that all share the same evidence, citation, and annotations. So that BEL nanopub is essentially a nano publication. We gratefully use that concept from the semantic web nanopub.org, and that is kind of a core piece of knowledge is a BEL nanopub. And that has the provenance, it has the experimental context, the annotations, etc. And we have a JSON schema that defines what goes in there and how it's structured. The BEL edge is the BEL assertion that has been compiled per se, compiled, computed, backbones, insertions, etc., into an edge store. So these are what we think of as possible biology that can be selected from to build specific hypotheses, i.e. networks, or views of biology in a particular context. Given lung cancer, stage 4, human, now I want to extract all of the BEL assertion, all of the BEL edges that have that context and start building a network from that. The API I'll sometimes refer to, if I refer to it without anything else, I'm talking about the BEL.bio API, and that provides BEL language, validation, parsing support, transformation, nanopub validation, and terminology services with equivalencing and orthologization. The abstract syntax tree I probably won't talk much about in this talk, but that's what we turn a BEL assertion into, and I am not staying consistent with the BEL statement there, but we turn it into a tree that we can then operate on in a computer science way. And this is something that Tony taught me a lot about, but apparently not enough, because when I tried to do it on my own, it was so not supported by BEL.bio. There will no longer be the knowledge assembly model where you take everything, you compile it into a set of potential BEL. We now have what's called an edge store, and we shifted over because the older system wasn't as scalable as what we're currently using, and we're currently on a native graph database. The OpenBEL API tooling, I don't know of anybody that is supporting it at this point, and I would recommend that that be deprecated at this point. The BEL scripts, Charlie is very happy to support those. I am no longer supporting a BEL script. If you don't know what a BEL script is, it's not a proprietary, it's a custom format for capturing BEL knowledge in a single text file. It is convenient for kind of a text editor, but it's not something that hardly anybody ever uses to actually edit directly, except for maybe Justina and back in the day, Natalie. Since it's a custom format, there are some things that have not been completely described as far as the definition of how a BEL script is put together, which has caused some questions from you, Charlie, I remember. And I have a love affair with Jason, and I'll share you the link to that article about it, but Jason I can load up no matter in every language I've worked with. XBEL is the XML version of the BEL script format. Again, we're not supporting that in BEL.bio. And the OpenBEL namespace equivalence files, there is some limited conversion into our terminology JSON files that we have, but it misses some things that are now necessary as far as the BEL.bio terminology services. So I am slowly converting the source databases that are found in the OpenBEL namespace files into BEL.bio terminology files as based on priority.
We're not supporting, just make mention we're not supporting BEL.1.
We are also, as part of BEL.bio, not supporting BEL.1. We will convert from BEL.1 to BEL.2, but we consider BEL.1 end of life at this point, at least for what we're supporting in BEL.bio. PyBel still strongly supports BEL.1. Pardon? Yep. Yeah. Yeah. And we consider it forward convertible very easily as well. But it's taken 4 years to get to BEL.2. We actually expect it to be another few months to get to the next version of BEL. Hopefully this afternoon session really accelerates that. So the BEL functionality is built on the BEL_lang Python module, and the BEL.bio API essentially almost completely depends upon this module to run, except for the API-specific code. It handles all the parsing, validation, canonicalization, and by canonicalization I mean when you have multiple namespaces where, such as the Human Gene Nomenclature Committee, HGNC, Mouse Genome Institute, MGI, and RGD, Rat Genome Database, those all have equivalences in EntreGene and SwissProt. So we consider the EntreGene identifier to be the canonical identifier it's configurable, but in most of our deployments, that's what we consider the canonical ID for all of the model organism database lexicons, names. You can configure that in the BEL.bio API. You can decide that SwissProt is your actual canonical identifier. It's up to you to decide how you want to do that. As far as BEL statements, we don't really care because they're all equivalents. We just need to have a primary identifier that would be unique in our database for the edge store. And of course, orthologization is built in, and we compute the edges from the primary BEL assertions. So an example is has member. So a complex has a member. That would be a computed edge in the edge store. It also provides a CLI so that you can do some operations directly in your text command line interface. So talking about multiple BEL versions, the OpenBEL API only handled one version of BEL at a time because it was the code base was built in to effectively the API. The way we've structured BEL.bio is the BEL language is determined by a BEL specification file, and that BEL specification file indicates what functions are allowed, what the parameters of the functions are. So if we drop in another BEL specification file, we can handle simultaneously another version of BEL. And there is no limit to how many versions of BEL that we can handle. It just depends on how many BEL specification files you want to deploy with your particular API. We have two ways of parsing. We provide what's called an eBNF parser descriptor, and that's generated from the BEL spec. We also have, for our BEL completion, it turned out that we really need to have a more flexible way of parsing, so we actually have a character by character based parser. It's more manually generated, and so we have both flavors. I'm not sure which way we're going to go in the future for handling that. And we're hoping that from version of BEL to version of BEL, we can come up with a way of describing the migration transformations from one version. So, for example, the substitution function in BEL.1 is now a variance inside of a protein or gene or RNA function. That migration transformation we're hoping to be able to describe inside the BEL specification, so then we'd actually have a way of migrating in a very straightforward way from one version of BEL to another in an automated fashion, and it would be part of the description of BEL. Any questions? Feel free to jump in. I also just want to provide a couple of screenshots of BioData Studio. This is powered by the BEL.bio API, so everything that is BEL specific in BioData Studio is the validation, parsing, BEL completion is powered by the BEL.bio API. It is in active use, which to me is always important for code. If it's not actively used, it tends to get stale. And we have also been striving for as much performance as possible out of the API. You can actually, we have a demo going of kind of a lot of the features and functionality of the BioData Studio in the back, and Anselmo will happily talk you through it. He's more conversant with the front end. I've been more focused on the back end. But these are the causal bionet networks that PMI was kind enough to contribute into the public domain. And we can demonstrate more functionality as far as manipulation and viewing of the networks, as well as the nanopub editing. We've actually had a great deal of encouragement, and I don't know if I can say who encouraged us to improve the nanopub editing, but the nanopub editing has gone through a lot of iterations, and we put a lot of work into it to make it as easy to find nanopubs. In this case, we're actually doing a PubMed query at the top, and when we do it, we find these 7 different nanopubs. So each one of these is a different nanopub. The evidence is highlighted if it can be found in the abstract between these brackets, these bars. And if we can't find the evidence in the abstract, it's listed down here. And then the assertions associated with these nanopubs are listed over here and color-coded, so you can match them up with the nanopub.
So it's showing up here that those texts were already curated, but they're being put in context of the full abstract?
Yep.
Okay, so it's not like you're given a fair...
No, we're not Belief. We're hoping to integrate with Belief technology at some point in the future. We do have PubTator that we can pull in to this for creating nanopubs. The more support we can provide for curation, the better. This is kind of the first pass, or I guess the second generation, but we have there's still several things that we would like to do to provide more text mining support for this process. And of course, everything that we do is also faceted if there are facets to make it easier to search, find, and curate. This is an example of the nanopub preview on the side. Take a quick look, and then if you want, you can go and edit from here, or copy. So we were strongly encouraged to have an easy and highly functional copy so that you could start curating a particular nanopub, PubMed ID, and then just keep kind of copying the citation, the evidence, or the annotations from one to another, and make that simple and customizable. Another big piece of what we have to do in supporting BEL is support the namespaces. And after looking at the different options and a lot of discussions with Natalie, thank you, as well as Charlie, it looked like it would make more sense to simplify the namespaces and put the onus on the API to understand what part of the namespace and how you wanted to use it. So our terminologies actually know what entity types. So there's two kind of flavors associated with the terminology. There's an entity type, and that says what type of thing can go into a BEL assertion at a particular point. So you can only have a protein entity type and a protein abundance as a namespace. You can only have a biological process in the biological process function. The annotation type, if it's a cell line annotation type, then they're associated in the terminology, so we can do filtering. So we do both filtering on the entity types and the annotation types as well as the species when we're providing namespace suggestions while you're building a BEL assertion. So in the Go biological process, Go biological process ID, so these are two different flavors of namespaces, effectively the same thing. One had an ID form, one had a name form. We're just trying to make the API smarter and get rid of a lot of the complexity of a lot of different namespaces. So all of these essentially map to Go now in our system. And we also spent a lot of time trying to figure out the workflow to make this easier, because our older system was very powerful, but it was also pretty complicated to be able to kind of drop in and help out. So one of our goals was to make this easier for other people in the community to develop their own terminology, collection scripts, and generation. So we have a template, and it's fairly bare bones, and that template script essentially says, okay, first thing you need to do is go out and grab the source database, pull it down, and compress it, and store it in the standard location based on your configuration of where these source databases go. So for this, we go out and pull Entregene, Swissprot, Kimball, Kebi, etc., and download them, store them. Then the next step is to process them into load files. So we don't actually have a monolithic system that grabs the terminologies and loads them into the database. We first generate these terminology files, which are collections of JSON objects. And JSON is it's a JSON script object notation format. If you're not familiar with it, it's a very programmer-friendly way of capturing things that you can read almost directly into a programming language data structure. We further capture these as there's something called JSON lines, where you can capture each JSON object as a single line in a file. I did this after I found I'd blown memory on my laptop because I was trying to read in the 21 million lines of, or 21 million concepts from genes from Entregene. And then I changed it to working on one at a time, and there's no memory constraint at that point. So there's a lot of thought and a lot of iteration going into doing this, but the goal is grab your source file, generate your JSON terminology files, and then the next step, which is completely asynchronous and disconnected, is tell the API, here is the URL to your terminology file, and it says, okay, and it puts it into a task queue, and then when it gets to that point, it goes out, grabs the terminology file, downloads it, and then loads the terminology up into the system. One additional thing I wanted to do as much as possible, especially for the customers of BioData Studio, is I did not want to have to take down the system in order to be able to load up a new terminology or update a terminology. So our focus was on making this so you could do updates on the fly without impacting any users. And we do that with Elasticsearch by essentially loading the new content and into a new index, and then mapping that new index into our terms index and getting rid of the old index for that. So there's kind of an instant switch from using the old to the new terminology, but there's no downtime in the system. Here is an example of doing a completion. So inside the protein abundance function, the cursor location here, and it could be anywhere in the BEL completion, wherever that cursor is, I know which function I'm inside of. I understand what entity types are allowed. Over here is the help. That is that's actually built into the BEL specification file, or generated from the BEL specification file. So we denote that we're inside a protein abundance. Here is kind of a shorthand version of what can be in a protein abundance. It can have a namespace argument in this situation, because we've already gone past the point where you can have a fusion as the first parameter of a protein abundance. You can have a location location and fragment, one of those, and a variant or PMOD, 0 or many of those. And then there's more kind of longer detail here. But the thing is, knowing where we are in the completion, knowing what kind of function, I also know what kinds of things are allowed. So I know that proteins are allowed in this case, and based on, again, feedback from our customer, we have tuned this so that depending upon where the AKT is found, it floats certain results higher than the others. We've also tuned we know before we even start at the very beginning what species we're working with, because that also filters what gets returned. And we have it configurable where you can say, I want these namespaces to be prioritized over these other namespaces. So there's been a lot of tuning to go into this to make it as easy and give you the best results possible. And it's also one of the reasons we decided not to go with OLS, the ontology lookup service, because we wanted to have a lot more flexibility in tuning the completion. These are some of the terminology statistics that we currently have in the system. And it's using Elasticsearch, which has been proven to be incredibly scalable and very, very fast. Those are, I have one in here, but it's down below the fold, SFAM. They're available. I just don't have it loaded on the demo server.
Do you find that people use those a lot in curation?
There's not a lot of people that use BEL. PMI is using it. I think. Pardon? Okay.
What I mean is that people, when they're curating, tend to want to express things in terms of AKT or ERK.
Yeah. Well, Natalie and I have been discussing trying to redo the protein family and make it more less Solventa or less custom and more open.
Okay. We should. Okay.
And that's a working group that we should probably, because you're absolutely right. And we should also talk this afternoon about how to identify things that we don't have a namespace for, a namespace identifier for. And because Mari and I were talking about that last night, and maybe using a UNK colon something, and then the label, then we could start, regular searches to say, are there any UNK colon labels that fit this entity type in this location that map to something that is now in our namespace? So we can put placeholders in, as long as we know that they're placeholders, and we know what kind of thing it has to be based on what function it's inside of. Then we can just kind of do continuous checks to see if anything shows up, and then float that up with a notification in whichever system, BioData Studio or other, that is using it. Yeah.
Yeah, we ended up curating it. I know the resources through on the television.
Oh, okay, that'd be great. Yeah. So, summary, BioData actually provides services for BioData Studio. We also have data stores for nanopubs, networks, and edges. And that's kind of separate from the BEL.bio part. We also provide support and maintenance around the terminologies as well as consulting that's BEL related. So just a little bit of shout out for what we're doing at BioData. All of this is heavily dependent upon a great community of people. Natalie is fantastic at sharing her knowledge and expertise. Anselmo, you see a lot of what the interface is all him for BioData Studio. David Chen helped in building BEL.bio, and all of this is built on the shoulders of Tony and Nick, who built OpenBEL.api. Because I, some cases, looked directly at the code and kind of translated it over to Python. But the lessons learned from that experience in OpenBEL API has really informed this. And then if you want some bedtime reading to put you to sleep, there are some, these will be shared so you can look at the links. My JSON philosophy is actually embedded in the article on Medium called My Love Affair with JSON. I can't sing enough praises of JSON and the simplicity of working with it. So, thank you. Any questions? Tony?
What exactly is the line drawing?
So, all of the BEL-specific functionality we have kept in BEL.bio. And the things that are related to kind of working with it in the BioData Studio would be the proprietary section. Now, in our plans are next year, I can say this, right?
I don't know what you're going to say.
About BioData Green?
Oh, yes.
Yeah. Our plans are next year, are, as soon as we can manage it, is to provide a public version of BioData Studio where you can put whatever you want in there that is publicly accessible. Use it as your notebook, whatever. We'll provide the ability for you, like GitHub does with their public versus private. If you want to keep your information private or keep parts of it private, you can pay a small fee per month or annually to have a private section. But we feel very strongly that the only way BEL will succeed is if there are tools that make it easy for biologists to work with BEL. And we are trying to figure out the commercial side of things so that we can support BEL for the world. Because in our estimation, you can't do biology unless you can talk about it. And we can't talk about biology yet. We're kind of stumbling around. Chemists have had the chemical reaction language for 100 years, and sorry, they are well ahead of us as far as being able to work with chemistry.
The stores that William also talked about, the Down Pub store, the Edge store, and the other stores, those are kind of white box right now, where we plan on releasing a version of them for everybody to use, so they can actually use the network store and the other stores as well. We're not quite there yet because we're moving pretty quickly here right now, so we're just making sure that things are lined up. But we plan on being very transparent, especially with the BEL.bio environment.
And there are lots of things on the BEL.bio side which would be fantastic if we had help with. There's a lot of things that are working well, a lot of things that are working good enough, and a lot of things that I really would like to work better. And it's just, as Anselmo said, we're moving really fast.
Well, do me a favor, just talk about scalability real quick for the new platform as opposed to the old one.
So we ran into about 1 million edges on a reasonably sized server, I think, Tony. We ran into, it basically fell over. I've tested the new database system up to about 150 million, and that was on one small server. The new, both Elasticsearch as well as ArangoDB are distributed databases, so it's been tested, the ArangoDB has been tested to at least 1 billion. Elasticsearch has been tested to 1 trillion documents. I don't think we're going to have any scalability issues in the near term. Kyle. Well, we're very eager to have if we can get a source of named entity recognition or prototypical BEL statements, we would love to have those in a kind of drop them in. You put in the PubMed ID or the document, and then it would drop in these entities that you could then easily drag into a BEL statement or set up as annotations. I don't have a good demo, but we could actually demo it where when we go and pull the PubMed ID, we also pull the mesh terms and the chemical substances from PubMed. We can also pull PubTator, though that's not implemented yet. But the mesh terms and the chemical compounds get brought up as soon as you put in your PubMed ID and you say, do you want to add these as annotations or not? Because it should be your choice. So that's the kind of thing that we want to provide as much curation support as we can. We just need to have a good source of those. Yeah. And that's what I'm hoping we can more tightly couple with belief.
You said you're not supporting CAMs.
No.
So when you look at things like the Alzheimer's disease model or the market, how does that get presented?
So the model would be a network, and that would be extracted out from the edge store. So the CAM and the edge store are essentially equivalent pieces in the two platforms.
What that model was?
Well, the dictionary, the network essentially is stored in the network store that we have, and that can be imported or exported in what we call JSON graph format. Oddly enough, another JSON data structure definition. But the
an edge store and a network store.
yes, and that's one of the reasons that we're trying to keep those terms as far apart as possible, because when you start talking about graph databases and graphs or the network database and then networks, when you have networks that have two semantically similar but distinct meanings, it just makes things impossible. So we talk about the edge store as a set of possible biology in a edge database or graph database, and then the networks to us are focused extractions for a particular biological purpose.
Is that more or less what a CAM is?
What I understood a CAM to be at the point that I came into Solventa, Genstruct Solventa, the CAM was a database of edges. But I understand in the earlier version of that.
In the earlier version we have what we call the knowledge repository, which is
Yeah.
your edge store. And then we would pull elements from that to make knowledge assembly models, which were individual networks to solve particular problems.
So your definition of CAM is more of a network, and my definition was more the
edge store sounds like it's all the knowledge I have.
It's all the knowledge.
Extract from to build a model.
Yep.
And the network is the model. So it's the same paradigm, just different.
And that's why I went through the glossary immediately, because we've over the last 12 years, 14 years of Solventa and even the Open BEL, we've gone through multiple, and I'm finally have to get rid of CAMs. Tony, and BEL scripts.
Is there a proposal for where the open source ecosystem is? Now that it's fragmented among all these places, BEL and
So BEL is a distinct. It has a different purpose, and
it's part of the ecosystem of tools.
yeah, we don't. We actually should describe where everything is, and I have been remiss in not at least linking from the BEL.bio website to everything. I think I point to PyBel
Yeah, I think you did.
on BEL.bio, but yeah. Okay, but we're trying to at least interlink. But there are different projects with different purposes.
The Open BEL, though, the GitHub organization is sort of dead. No one's really working on that.
Yeah.
So
At some point. Yeah, so we should probably go and I haven't felt like I had the right to go and say put deprecated on everything.
So with OpenBel, I mean, so what is the role of OpenBel? I mean, I thought that was going to be the...
As an organization or as a technology base?
And also, I guess, a legal structure, right? I mean, OpenBel Foundation and maybe...
Keith?
Is there an open bell foundation?
Right? I mean, within yeah, okay.
There's no OpenBel Foundation anymore.
I think the key is can you find a sponsor of something like
Oh.
either grant funds or corporate,
Yeah.
Basically, Applied Data Solutions, which was supporting all of this development beforehand. Now we have BioData. We're basically BEL.bio has corporate sponsorship. So BEL has corporate sponsorship, which is something that I think is needed to some degree for a long time now, because we're actively funneling money into the development of BEL, to the retooling of the platform. We're going to be putting meetings together with people to continue to extend the language and bring it out. So
who owns the BEL language right now?
That is a really good question.
BEL was OpenBel,
And
originally that was supposed to be open source.
and something that we're going to talk about this afternoon, and how to evolve the language with the community. I was, and Ted was, Ted has to step out. Ted and I were intimately involved in trying to get grant support, funding support for the OpenBel Foundation. After that experience, I decided corporate sponsorship hopefully was going to work a little bit better.
yeah, grant support for open source.
Yeah, it's really painful. And pharmas aren't really that into giving you blank checks for whatever. I've been impressed with how well you've done, Keith, at getting pharmas to open up their pocketbooks.
Let's be aligned with a particular problem.
Yeah.
Very attention to
I've taken up more time than I should, especially as the timekeeper. So we are now up for lunch. If we can all be back at, what time did?
at 2.
At 2. If we can all